Hello, ladies and gentlemen. This is your favorite program of science, The Power of Nature. Sadly, we can't be together because of the coronavirus, and we have to stay in our houses. But we are going to have this meeting by Zoom. I'm Dr. Carlos, and today we have a special program with four of the greatest scientists of the entire world. Please welcome Dr. Armin, Dr. Betsy, Dr. Kevin, and Dr. Jennifer, the team that won the Nobel Prize of Chemistry in 2009. They had a huge investigation about the structure and function of the ribosome. Today, we are going to talk about their investigation and how this knowledge can help us to understand more about ribosomes. To start to talk about ribosomes, we have to remember that we are here thanks to the DNA. What can you tell us about the DNA and its relation with the ribosomes, Dr. Armin?
Well, I'm going to start giving a little reminder of the function of ribosomes. It all starts in DNA because the genetic information in living systems is stored in the genome sequences of their DNA. This genetic information is carried out by DNA polymerase, so each daughter cell can receive one genome copy at every cell division. The protein production process begins with the transcription of DNA into mRNA that is carried out by RNA polymerase. Then translation of mRNA is carried out by the ribosome. The mRNA sequence is decoded starting from an AUG codon followed by a sequence of codon. And finally, a little reminder of the components of the ribosome. It basically consists of a small 30S and a large 50S subunit and have three binding sites, the A, P, and E sites.
Thank you very much, Dr. Armin. So, Dr. Jennifer, what could be wrong with these ribosomes?
Well, it's known that the ribosomes catalyze two chemical reactions, steps involving covalent bonds, peptide bond formation, and ester bond hydrolysis during termination. Something important during the elongation phase is that when the tRNA cognates something important to an amino acid encoding A site, codon must be efficiently selected. Some mistake could happen during the elongation will generate a reduced activity or alter of synthesized proteins. And the second, to greatly reduce of the ability to produce breeding-mate proteins. The chemical mechanisms of the covalent reaction steps carried out by the ribosome is still unknown, and how the antibiotics and ribosome mutation can the codon be read out and down. And that's all.
Thank you very much, Dr. Jennifer. And before we continue, I have to tell you something really important. I have to talk about the ribosome and the subunits in high resolution and the impact that this can have in the future when we look to new drugs. One of my best friends, Jonat, a famous scientist who had made many articles, worked with Dr. Armin, and they had many significant contributions to obtain a high resolution structure of the 50S subunit of the ribosome. They used crystal from Alacura Maristui, and Dr. Armin and Dr. Ada have been struggling to obtain a high-resolution structure of the 30S subunit of the ribosome. In the recent publication, they said that it's really hard to obtain it, but they are working hard on that, so we hope to get a high resolution of the structure of the subunit 30S soon. To make this possible, they work with cryo-electron microscopy reconstruction of the ribosomal 50S subunit from Dr. Frank in 1995. So guys, to get a high-resolution structure took since 1995. This technique that Dr. Frank used let Dr. Armin and Dr. Jonat get a low-resolution structure of the 50S subunit. Everything that we know about the structure of the ribosome started here. Dr. Frank's reconstruction was in low resolution. It displayed right-handed double helical density typical of a form of RNA. It demonstrated for the first time that the phase problem was tractable for ribosomal subunits and even the whole 70S ribosome. This strongly suggests that both high-resolution structures of the ribosomal subunit and the whole ribosomes were within near reach. 1 year later, a middle-resolution structure of the 50S subunit was reported by Dr. States. The same year, a low-resolution structure of the 30S subunit was reported by Dr. Jonat, but the problem was that neither one of these structures were not high enough resolution to complete anatomic models, but they provide the necessary stepping stones on the path to get the high resolution structures. In 2001, Dr. Noller reported the crystal structure of 70S ribosome, but a structure of the whole 70S high resolution was first obtained by Dr. Kate for an empty ribosome from Escherichia coli. After the scientists could obtain high resolution structures, a radical change of the ribosomes research had occurred. The structures of the two ribosome subunits provide new information in the structural folds of RNA and the binding profiles of antibiotics. This is very important for the future investigations. What do you want to tell us about this, Dr. Betsy?
Okay, so just basically to sum up what we all have said, the thing that we are investigating here is that DNA cannot leave the nucleus. So DNA contains all that we are. So in order for us to express that, we need to take the information out of the nucleus. So that's for what the cell uses, the transcription process. So this information goes out, but the way we're gonna work is with proteins. So the thing about proteins is that they need to be created from the DNA. So we take DNA to RNA, and then the RNA, well, the messenger RNA goes to the ribosome, and then the ribosome divides them by codons. Let's say a codon is a team of three nucleobases. So it could be like adenine, thymine, or guanosine, something like that. And then it translates that into amino acids. And these amino acids will constitute a protein. The thing is that if this protein goes bad, something can happen in our body, for better or for wrong. And this is the basis of our investigation because we are not the only ones who use this process to make proteins, also bacteria do. So if we target this to the bacteria, we can have antibiotics that mess up with their proteins process, and in that way we can stop infections. So that's the real I don't know, the real approach of our investigation. And we have investigated more about that. For example, the relationship between the codon and the anticodon, which is what Dr. Kevin will talk about.
I just want to establish a couple of points related to the accuracy of the translation of the mRNA. For an accurate translation of the genetic code, this process depends on what is called the standard free energy difference between the codon and anticodon pairs. This is important because this way the ribosome can enhance the accuracy and thereby reduce the frequency of amino acid substitution errors. Another thing that I want to mention is that the genetic code is redundant. So there are 20 canonical amino acids, but there are 61 codons, all which are used in mRNAs. One solution to this problem is provided by the existence of several iso-accepting translation RNAs for the same amino acid. Also, the accuracy of the codon reading on the ribosome can be tuned up or down just by mutation on the ribosomal RNA and ribosomal proteins. This is exciting because there are some ribosome targeting antibiotics and drugs that have profound effects on the accuracy of codon reading. Thank you so much for listening.
Okay, so Dr. Carlos, thank you for having us here. It's really important for us, for people to understand what we're doing and how this is so important for the advance of science and medicine. So we are very grateful for being here. And thank you to your program. And I hope everyone enjoyed what we have presented here.
Thank you for invite us, and it was an honor.
Thank you, doctors, for coming to my program. It was an honor to have you here, and it was a good idea to let people know about the ribosomes. So thank you very much, and I hope you to have here you soon.